for that kind introduction. I was going to make fun of all the people that showed up at 9 o'clock to come watch chemistry seminars, but there's only like 5 people in here, so I'll skip that part of my intro. Today I will be talking about the cloning, purification, and characterization of human aldehyde dehydrogenase, Aldh5A1, and its ability to catalyze the detoxification of certain chemotherapeutics and the toxification of environmental contaminants. My name is Todd Hoffman, and my advisor for this project was Dr. Sri Rama. And the title is really long. Let me digest it for you. My project consisted of cloning, purifying, and characterizing this Aldh5A1. Aldhs are known to detoxify these chemotherapeutic drugs that you take if you have a tumor. These drugs you take are very harmful to this tumor cell, and these few Aldhs are known to detoxify that effect, and it's also known to detoxify some environmental contaminants. So that's what I'll be talking about today. First of all, what is an Aldh? An Aldh is an aldehyde dehydrogenase. You can see where it gets its name from. This is a general reaction that it catalyzes, and as its name says, it's a dehydrogenase. So it takes this aldehyde and converts it to this product. You can see it gets rid of this hydrogen, so that's where it gets its dehydrogenase from. Normally there should be a carboxylic acid, but these are present in solution, so this H just falls off due to the water that is present. And this is a proposed structure of Aldh5A1. Well, an Aldh is an enzyme. There's actually a family of enzymes of Aldhs, and they catalyze the oxidation of aldehydes, which was that previous reaction that I showed. There's at least 17 different genes that code for all the known Aldhs. They're found all throughout your cell, in the cytoplasm, in mitochondria. They're also found in many different tissues, your eyes, liver, skin, stomach. Basically, anywhere you're going to have an aldehyde, you're going to find an aldehyde dehydrogenase, which is pretty ubiquitous throughout your entire body. So, why do we need these things? Well, when you eat such things as proteins, they break down amino acids, fats into lipids, and then sugars and starches into your carbohydrates. And when you metabolize these, they produce aldehydes. And aldehydes are very reactive molecules within your body. A lot of them are very toxic, so you need these Aldhs to break those down into the corresponding acid. But the most familiar pathway for most of you might be that of ethanol metabolism. So I'll ask the audience, does anybody know anything that you eat or digest that has ethanol in it?
Beer.
Correct, beer. Yummy. Well, correct. Anything, any alcoholic beverage, actually the alcohol that's in there is called ethanol. And when you drink this, your body breaks it down via alcohol dehydrogenase into the acetaldehyde here. And the acetaldehyde is actually very toxic in your body, and that's what's responsible for giving you the buzz, which makes you feel good. However, if you have too much of it, you get your hangover, you don't feel so good. Maybe you feel like your world is spinning around if you've had too much, or perhaps you look like this polar bear afterwards. I know I look like that instantly on my birthday. But what your body does with this acetaldehyde, the Aldhs that you have in your body actually break it down into acetic acid, and when it does this, you feel much better, happy like this smiley face guy. And actually, the Aldh that's responsible for that is Aldh2. And as I said before, aldehydes are incredibly reactive molecules. A lot of times they're bad. However, there are very good effects of retinaldehyde, which is an aldehyde. This is found in your eye. It's in the cis form. It's coupled with an opsin protein, and when light hits it, it straightens out into the transform of the retinaldehyde. When it does this, it gets out the active side of that opsin protein. It's too big, so the opsin protein releases. And it starts a cascade effect of chemical signals that eventually get to your brain, and it interprets it as light. So that's a good effect. However, many of them are bad. Can be cytotoxic, cause mutagens, and cancer. Just a few aldehydes. Acetaldehyde, which was from the ethanol metabolism, it's toxic. It's a probable carcinogen. Formaldehyde is toxic. Most of you probably recognize that it's used in disinfectants. It's also what preserves cadavers or dead bodies, and it smells really bad when you smell it. You're smelling the formaldehyde. And how that works, it actually takes the primary amine groups in your protein and irreversibly crossing them with nearby nitrogen groups from your DNA or protein, and that actually preserves your tissues then. Butyraldehyde, glutaraldehyde, also very highly toxic. Glutaraldehyde is used in the sterilization of hospital equipment, also it's used for many things, industrial water cleaner as well. So you can imagine if you have these aldehydes and your ALDHs aren't working properly, they're dysfunctional or you completely lost it, it can cause complications. A few of those complications are an Aldh2 polymorphism. The Aldh2, if you remember, was involved in the ethanol metabolism. So one amino acid change in this Aldh2 protein leads to extremely high sensitivity to alcohol. Basically, if you have this polymorphism, those people cannot drink alcohol at all. Otherwise, they'll feel like they're hungover as soon as they drink it because they cannot convert that acetaldehyde into acetic acid properly. Can be kind of beneficial because the people who have this, obviously they get sick every time they drink alcohol. So people who have this have an extremely low ability to get alcoholism. And then an ALDH3A2 mutation can cause Sjogren-Larsen syndrome, which is a thickening of your skin, becomes fleshy-like. People with this also have mental retardation and their legs are spastic. ALDH4A1 mutation causes type 2 hyperprolinema, and that is characteristic of mental retardation as well as convulsions. And then the protein that I'm studying, ALDH5A1, a deficiency in that causes 4-hydroxybutyric aciduria. I'll be talking about that in a little bit. So a little bit more about ALDH5A1. It's also known as succinic semi-aldehyde dehydrogenase, not too important to remember that, SSADH. It's involved in the metabolism of GABA, which is gamma aminobutyric acid. That's a structure there. And GABA is a major inhibitory neurotransmitter in your body. When you have this present, it keeps your neurons from firing. And you can imagine if there's any complications with ALDH5A1, since it is involved in the metabolism of this inhibitory neurotransmitter, people that have complications that have convulsions, seizures, things like that. ALDH is also the predominant oxidizing enzyme for 4-hydroxytrans-2-nonenol. The structure is here. It's very highly toxic for your neuron cells and it's accompanied with numerous neurodegenerative disorders. Again, deficiency in this ALDH5A1 results in 4-hydroxybutyric aciduria, characteristic of delayed development. People have hypotonia with the deficiency. Hypotonia just means a lack of muscle tone, or should be a lack of muscle tone. Basically, they're going to be very weak. Myopathy with ragged red fibers. It's a neuromuscular disorder, so people maybe won't be able to use all of their muscles since some of the neurons won't be able to fire. Other characteristics: abnormal behavior, ocular abnormalities, mental retardation, and again, since it's involved with this GABA, people have seizures, convulsions, things like that when they have a deficiency in the ALDH5A1. Well, why is this research important? I kind of went over aldehydes and ALDHs, what they're used for. Well, ALDHs, as I said at the very beginning, oxidize that aldehyde reaction, and there's aldehyde intermediates produced during the metabolism of those drugs that you take. These are two commonly used drugs, cyclophosphamide and ifosfamide, and when you take them in your body, I'll show you in a little bit, these actually are very toxic eventually when your body breaks them down, but ALDHs undergo a different pathway when it gets a hold of them and makes them non-toxic. And this is the pathway for that. I'll explain a little bit. You can see cyclophosphamide here. When you take that drug, it's ingested into your body, it can go one of two ways, via these P450 enzymes. We'll just go this route first. Produces this dichloroethyl chlorophosphamide plus this chloral acid aldehyde. And again, it's an aldehyde, causes nephrotoxicity, which is toxicity to your kidneys. And what this is, since it's an aldehyde, it can be acted upon by an ALDH with NADH+, and goes to this chloral acetate, which is non-toxic to your body, just gets excreted through the urine. The other pathway it can take is to this 4-hydroxycyclophosphamide, spontaneously undergoes this ring detemerization to this aldophosphamide, where it can go one of three directions. The direction you want it to go if you're taking it if you have a tumor is down here to this phosphoramide mustard, which is very toxic to your cancer cells and ends up killing your tumor, which you want. However, if ALDH gets a hold of this, produces this carboxyphosphamide, It's non-toxic to your body, also gets excreted through the urine. If you have a tumor, you definitely don't want that happening. And another route it can go via just a base reaction with water can go to acrolein, which is also toxic, causes atherosclerosis, which is a hardening of your arteries, diabetes, cancer-causing agent. And again, this is aldehyde, so Aldh can take care of it, produce acrylic acid, and then eventually go to malonate. And the other part of the talk was detoxification of some environmental contaminants, and those contaminants are ethylene glycol ethers. They're used as industrial solvents. They're found in dyes, inks, so if you work at a printing press or something, you might be exposed to these, and the ink that you're using are found in lacquers, paints, basically your industrial solvent. And ethylene glycol ether alcohols, that's the structure of them. In your body, they get broken down into these aldehydes, and again, these aldehydes can be very bad, and the aldehyde dehydrogenases in your body get a hold of those and convert them to the acid form. Here's the pathway for this. You can see the ethylene glycol ether here. It's broken down by an alcohol dehydrogenase into this aldehyde form, where then it is acted upon by the ALDHs into 2-alkoxyacetate. This can be broken down over here into 2-alkoxyacetyl coenzyme A, which can get broken down into acetyl coenzyme A, which is actually a precursor molecule for the Krebs cycle. It's very important. Krebs cycle is where we get a lot of our energy from. Also, fatty acid synthesis. So that's the main pathway that we're concerned with, the ethylene glycol ethers. The toxicology of these contaminants comes from animal studies usually, or for the most part, human toxicology is limited just to case reports. And for time concerns, I'm not going to go over all these, but you can have acute toxicity when you're just exposed to it every so often in the environment. It causes hemolysis, which is bursting of your red blood cells, basically. Decreased white blood cell count. White blood cells are used in your immune system, as you know, so you do not want that. Also, hypercellular bone marrow, which means a reduction of the cells in your bone marrow. And this causes incidence of leukemia. Metabolic acidosis can be caused when you break these down into the acid form. Esophageal, stomach, and other GI tract tumors are very common in the animals that are exposed to these ethylene glycol ethers. If you're constantly exposed to them, let's say you work at possibly a printing press and you work with this ink that has them, neurological symptoms occur, become lethargic, have lots of memory loss, headache, dizziness, cause teratogenic effects, which are basically birth defects, spermatoxicity, again, hematoxicity, which is like the bursting of your red blood cells. The 2-methoxy exhibits the strongest and broadest spectrum of toxicity, which is this first one here, followed by 2-ethoxy and 2-butyl, which are these two, and this fourth one here. So these top two and these fourth ones are the ones that are the most toxic in the body. And again, why this is important, throughout this whole reaction, this ALDH step is actually the rate-limiting step, so it can only go as fast as this ALDH will convert it to the acetate form. So if it's not very fast, these aldehydes will build up in your body. It becomes very toxic. So now that you know a little bit about all that stuff, what were the goals of our research? Well, we want to clone, isolate, and characterize this Aldh5A1. And we want to do that because these reactions, such as this right here and this one, these ALDHs are known to do all these, but it is not known whether Aldh5A1 plays a role in any of those. And we're going to test if it plays a role in the detoxification of those chemotherapeutic drugs, and also if it plays a role in the detoxification of those ethylene glycol ethers. So here's a flow chart that hopefully you can follow along with me when I go through this. I know it's a big problem for me when I'm listening to people's lectures, trying to follow along with the steps that they're going on in their research. So I'll be coming back to this slide numerous times. Hopefully you can keep along. In the first step, and there is the growth of Aldh5A1 E. coli clones in selected media from a purchased stock. So when I started this research project this summer, we had tubes that looked very similar to this that had E. coli growing on them with a T blue script R vector insert, which contained our human Aldh5A1 gene in them. What I did is take sterile loop and just scrape one of these, and then put it on an LD amp auger plate, just swab it right on there. Grew it overnight, and when that happened, the plate looks similar to this. It's actually not a plate that I grew, but the plate would look very similar to this. And what you do, take one of these individual colonies and plop it into the liquid broth form. And the reason you take just one colony is because obviously bacteria grow asexually, and what that means, they just divide in their daughter cells and they have the exact same genes that they do, which means once they divide and grow and grow and grow, eventually, if they do it enough, you'll see a colony there, which are all genetically the same. Take that and grow up in here, and then they'll grow even more of them. So how do we know that they're clones? Well, as I said, when I first got these, they contain this P blue script R vector insert, and that's what this is here. And for those of you who don't know what a plasmid is, you can think of it as a circular piece of DNA, which is actually what it is. But it's kind of man-made, and it contains these goodies that we can exploit here. And this P blue script R in particular contains a gene sequence that will code for ampicillin resistance, which is an antibiotic. So when this plasmid is inside these little E. coli like this picture, obviously plasmid's not going to be that big compared to this E. coli, but when that plasmid's in there, it'll give it a resistance to the ampicillin. When you grow it on these plates, you put ampicillin on the plates. And in theory, the only thing that should be growing on there, since E. coli don't have a natural resistance to ampicillin, the only thing that should grow on there is the E. coli that contain this vector insert, which also contains your Aldh5A1 gene. So the next step would be to isolate that plasmid that you grew up in that liquid culture. We did that. There's several ways that you can do that. The easiest way for us is just to purchase a kit from any biotechnology or biochemical company. And Sigma Aldrich has a really good one. It takes like 15 minutes. It usually works very well. But for some reason, with this project, the plasma mini prep kit from Sigma Aldrich, we used it 3 times, and for whatever reason, we didn't know it did not work. So actually, another person in the lab was having the same problem, and he said it worked fine using this Promega kit. So we switched to that, and that was used successfully. I'll show you the results from that. Basically, the next step to test if you have that plasmid extracted is to do a restriction endonuclease digestion. And that will, it's a good way of testing if you isolate the plasmid. It's also a good way of getting your gene of interest out as well. So we originally tried cutting with the BAMH1 and XHO1, which are restriction enzymes. And what they'll do is cut your DNA at these specific sites. You can see BAMH1 here and XHO1 right by each other. And they'll cut the DNA, and in theory, you should have your gene of interest in one band, and then this rest of this vector, you should see another band. But we saw 3 bands, which means it's cutting somewhere else. So what I did is looked up the gene sequence of Aldh5A1 online, and sure enough, BAMH1 actually was found in the Aldh5A1 gene sequence. So BAMH1 is cutting our gene, which you don't want if you're trying to clone your protein. So we use KPN1 in place of BAMH1. Aldh5A1 does not have a restriction site for that. And we ran an agarose gel on that. Spare the details, but basically we ran this on this gel. You can see it's not very pure, so the plasma mini prep that we did was kind of contaminated. So there's another way you can get a hold of your Aldh5A1 gene instead of using this restriction enzyme to digest. That was not very pure. We tried to amplify our gene using a thing called PCR. PCR is basically the over-glorified version on any CSI show you're going to watch. That's what they do if they get a DNA sample. They run PCR. And I'll just explain what that is really quick for you guys. You take, in theory, you could just have one strand or one piece of DNA, and you take that and you heat it up really warm to 94 degrees Celsius, which is pretty close to the boiling temperature of water. 94 degrees Celsius will denature this, which means your double-stranded helix on your DNA will split into 2. And when that happens, you cool the temperature down. In our case, 50 degrees Celsius worked the best for the primers that we use. So when you cool this down, your primers will bind to the 2 DNA strands. And what primers are is the complementary base paired nucleic acid sequence to your Aldh5A1 gene or whatever gene you want. And it will bind specifically to that gene. And when that, you cool it down for a little bit, you warm it back up, and enzyme that is found in bacteria that grow in thermal vents. You know, they have to replicate their DNA. You take the enzyme that's responsible for replicating their DNA. After you warm this up, it finds these primer sites, and basically it'll find that, and then it'll sequence, or you'll have 2 strands after that of your gene, and you just repeat that cycle, warm it back up. Those 2 strands will then split into 4 and so on. So from 1, you go to 2, 4, 8, 16, 32, and so on. And after 30 cycles, that if you do the math, you get millions and millions of copies of your gene. And we did that, and then when we ran this on the gel, you can see in lanes 1 and 3, we have DNA product, and then 2, we have marker. And let's see if I can dim these lights. I don't know if you guys can see that really well. There's a band here, and then there's a couple bands up here in your DNA product, which you really wouldn't expect when you run a PCR reaction. You wouldn't really expect that in a PCR reaction, so that's not very pure as well. So we tried using a purification kit for PCR, and when we ran that on a gel, you can see again more than one band, which you don't want. When you do the next step, which is transformation, you want your specific gene to be in there. You want it as pure as possible. But it's not the end of the world. You'll just have to do a little extra work on the next step, which is the transformation of these Aldh2 genes into a host cell. And currently, that is where I am on this project. I've been actually working on this transformation step for like the last 4 months. Just for some reason has not worked. But what you want to do is take your Aldh2 gene, maybe from PCR, and you want to ligate it into your expression vector. The one that we're going to be using is PET44EKLIC. That's the one I've been trying to get it into. And then once you have your gene of interest into your vector, it can be taken up by competent E. coli cells, and then again, this is going to have resistances to antibiotics, and basically what I said before, you're going to have to grow colonies on a plate, grow in liquid media, isolate the plasma, and then test again to make sure that the ones that were growing on there contain an Aldh2 gene. And that's currently where I'm at. So since the product was not pure, I'm going to have to do all this, and so far, after 4 months of doing this over and over and over, these clones have not showed the Aldh2 gene in them so far. So the rest of my talk will be talking about what I have to do once this does work, which is grow that host cell in the liquid culture. So once that transformation shows to be successful, you can overexpress the Aldh2 protein in these E. coli, and you do this by plating them again on a agarose gel. This time, that PET44 vector we're using has resistances to ampicillin and chloramphenicol, so we're going to use that on the plate. It's also going to contain IPTG, which allows for the overexpression of the gene of your interest, and then XGAL, which is basically a control mechanism to make sure that the E. coli that are growing on here are ones that are overproducing your Aldh2 protein. And so you're going to have a bunch of these little E. coli overexpressing your Aldh2 protein. You pluck the colony, put it in there, and grow a lot of them, so you're going to have a lot of these bacteria overproducing your protein. As you can imagine, there's going to be a lot of your protein in there. So the next step would be to isolate this protein, and to do that, you obtain a slurry of the cell lysate, which basically means you centrifuge this down. A centrifuge just spins around really fast, so the cells that are in there will go to the bottom of your tube that you contain them in. The liquid will be on top. You can get rid of that liquid, add in a buffer that's going to break open the cells, a lysis buffer, and then you subject them to sonication, which is just a probe that you stick in your solution, vibrates at a really high frequency, and again, that's just going to bust open the cells even more. So you're going to have a solution that contains just a bunch of your protein and everything else that was in there from the cell, and you're going to run an initial test to test if your protein is in there. You run enzymatic activity using spectrophotometry. If you show you have activity, then you can start running all that lysate through a PD10 column, which is a desalting column. This is a buffer we're going to use for that. It contains 5 millimolar imidazole, which I'll get to in a little bit. But that PD10 column gets rid of a lot of the other junk that's within your cell lysate, and you collect fractions of these, and then these fractions you can run through a nickel-chelated separose affinity column. Using a his tag, the protein will bind to the beads within that affinity column, and the theory is that everything else but your protein will fall through this column, and your protein will be bound to the beads in there. And after you've washed it thoroughly and you think that everything else is out of it, you increase this concentration of imidazole, which binds to the beads stronger than what your protein does, and your protein will crash out into your fractions, and you'll have a purified form of your protein, hopefully. And when that happens, we're going to test the enzyme activity and all these reactions that I went over before. We're going to test them spectrophotometrically, at 340 nanometers. This reaction here with the NAD to NADH that produces light at 340 nanometers, and that's what we're going to utilize in these reactions. And actually, the reaction for this to also use NAD to NADH. And for the ethylene glycol ethers, we're going to test all of these. He wants to test all of these and see if Aldh5A1 plays a role in these, as well as this chloral acetaldehyde to chloral acetate, this aldophosphamide into the carboxyphosphamide, and the acrylic acid, or the acylane into the acrylic acid, and that's what we want to test. So just a summary of all the results. The P blue script R plasmid was isolated using that Omega kit. It could not be isolated for whatever reason using the Sigma kit. And that isolated 5A1 gene that was kind of contaminated, we transformed those into the Rosetta cells. Those transformed cells were tested for the presence of Aldh5A1 gene, and so far they've been proven to be lacking that gene. So the future research that we have to do is to get the Aldh5A1 gene ligated into the PET44 EK-LIC vector, which is an expression vector, and then transform that into the E. coli. Overexpress our gene so that it generates lots of your Aldh5A1 protein, and isolate and purify that protein and test for the enzymatic activity with respect to the oxazophosphorin chemotherapeutic drugs and the ethylene glycol ether metabolites. Acknowledgements, Dr. Sri Rama obviously is my advisor on this project. He provided all the chemicals and things like that, advised me where to go, provided the protocol and everything. And then Rainer Grant, Greg Leggett, an awesome pitcher, were also there when I started in the summer. And when we were doing our research throughout the summer, everybody was kind of making solutions for one another. And I'd just like to acknowledge all the solutions that they made and that I used. Questions?
Any questions?
I have one.
Yes.
Maybe you said it and I was just writing, but back when you were talking about amplifying the gene with PCR, why was temperature important? Like, why did you have to do it at different temperatures?
Oh, the cycle you're talking about?
Yeah.
When you heat it up, basically this will denature the protein and your primers will bind. And at this temperature, they melt. You cool it down for 30 seconds, which allows these primers, which are sequence specific for your gene that you want. When you cool this down, the primers will bind. When you warm it back up, the tag DNA polymerase, which is that enzyme that comes from those bacteria, will be able to elongate this strand, and then you'll have your Aldh1 genes. And what you do is heat it back up to 94 degrees. That'll melt those two strands that you have into four, and so on.
Okay.
And we use 30 cycles, so you'll have lots of your gene after that.
Dan, you have a question?
Yeah, so on the step that you've been having trouble with, what's going wrong?
The protocol that I was following actually was incorrect, and I think we recently found out what we have to do to get this Aldh5A1. The ligation step is basically what was missing, and it wasn't working. So.
What, do you know what it was doing?
Yes, actually, I was following an incorrect protocol. So instead of ligating into this vector, the protocol I was following just said you put your PCR product in there, and PCR product can go directly into your E. coli. You need it in this vector.
Okay.
That was okay.
Can you tell us why PCR product cannot be transformed straight into, why did that fail?
Well, it's a linear piece of DNA, and when we use chemical form to make the cells more permeable to DNA. They're not naturally just going to uptake DNA. And this gene segment is going to be really long and linear. It's going to be really long. And this vector, PET44, I don't have a picture of it, but it's going to be something like this. It's just a circular piece of DNA, ends up being a little smaller, and that can be taken into your cells easier. Yes.
Actually, when you're going to go on and test on the enzyme activity, are you going to be looking at just if there's some interaction, or are you going to be looking at rate? So you're going to be able to tell us something about how those steps work and how important this Aldh5.
Well, we're going to see, first of all, if it has any enzyme activity with respect to any of these reactions. And if it does, then we're probably going to go through and check, do enzyme or kinetics of it,
Okay.
and see how fast it goes.
So first you're going to isolate each reaction, and then you're going to look at the kinetics.
From what I was told, he has these products, and then what was that in our enzyme?
Okay.
See if it has an activity.
I have a couple questions on about 5A1.
Yes.
Where is that localized and why have it? I mean, how many isoforms are there for aldehyde dehydrogenase?
There's 17 different isoforms.
So why do you need 5A1? Why can't the other 16 pick up the slack?
Well, are you talking in regards to this reaction?
In general.
In general, I don't know. They're specific for certain reactions, and this ALDH5A1, actually 3A1 and 1A1 are known to undergo this reaction. And we're wondering if 5A1 does. We're not sure if it does or not. And it might be that all 17 could undergo this, but not too sure if it does or not. So we're going to use 5A1.
Were you involved in designing the primers for PCR?
Yes, I was.
And where do they bind relative to the gene, the 5A1 DNA?
What do you mean?
Well, you've got a gene sequence for 5A1.
Yes.
And the primers are going to bind
It's
around or in the middle or in it, or?
It's, let's see, we looked up the gene sequence and we looked at where the first start codon was for the gene. And then we had our vector that we're gonna ligate it into, so we combined that so it would stick into our vector. And then you have the first part of the start codon and the first part of the sequence from there, and then the stop codon for the first reaction.
So you had the upstream primer began right with the start codon? Is that what you're?
The primer didn't start right with it. It had a sequence for your vector that you're going to put it into initially, and then so it could bind with your vector, and then it has sequence-specific stuff for your ALDH5A1 gene that will allow it to bind with the gene as well.
How would you prove that the DNA that was sent to you was the DNA you wanted? I mean, you're assuming.
Yeah, you can do that with PCR. I mean, if you looked at a reaction, it didn't show to be very pure. It's got other bands in there.
But I mean, how would you prove that the plasmid DNA that was sent to you was expected it to be?
You could sequence the DNA of it, I suppose, if you wanted to. This obviously has a specific DNA sequence to it, and if you wanted to, you could run PCR on that as well, amplify it, and then do a DNA sequencing.
A simpler way that it wouldn't be as definitive as sequencing is just to do a restriction map. And you actually already have in your initial one, using the VAMH1 and the ZIL1.
Yep.
And what mass, if you added up and the masses of the one, you said you have 3 fragments.
Yeah.
What do those add up to? Do they add up
I
to-
don't remember off the top of my head, and I couldn't find the picture of this one with the 3 bands. I don't remember off the top of my head where that research enzyme site even was. But
So you don't know if they added up to what your
no, I didn't.
subtotal.
No, we were just looking to see that I had 2 bands in it. So we looked to see if I had a research inside of it, and it did. But, yes.
It's a dirty, rotten question. I just want to see if you remember.
Bring it on.
You were talking about the isomerization of cis and trans retinol. Do you remember why that happens?
Why that happens?
And why it happens so quickly?
The cis strand is actually not sterically favored, so it's going to want to go to this trans version. This is steric hindrance. This group is getting a little too close to these hydrogen stuff on there. It's more stable when it's in this form. So this reaction actually happens within 200 femtoseconds.
Okay, and do you know what happens to the cis when light hits it that makes it jump into the train? You know this, I just want you to think about it. Something special about that pi bond, right?
Yeah, you got radicals in there, and then
What is it, kind of, what is the light going to do to that, the electrons in that pi bond?
or it'll jump them up into the next energy state.
Okay, and why does that make it want to, so early in the morning?
Oh man.
Yeah, you're right, I mean, so it goes up to goes from a pi bond to what's the next state?
The sigma, or
Molecular orbital theory. You got it.
it's been a while. Not sure.
It's the pi star,
That's right.
and then the lobes don't overlap, so they have to flip before they can come back. It was a long time ago, I know. I just-
Any other questions?
I have another one.
Yes.
On the gene loop versus omega, I know they don't tell you what's in the reagents you're adding. You know, maybe let's just say, washcloth, one, whatever. But were the sequence of steps the same?
Roughly, the wizard, in fact, it was from, Omega actually had a step where you had to run it through a filtering column first, or during a step you had to run it through a filtering column twice, where this didn't necessarily use that, it used a smaller version of it and just a microcentrifuge tube, where this was using about 30 milliliters of your solution.
30 milliliters of the overnight?
Yeah, of a buffer, or I can't remember what it was, it's a resin that will bind into the filter that you put it through, and this didn't necessarily have that.
Renata, any questions?
Yeah, right. I'm just here to be my best student.
Are there any other questions? Well, fantastic.